Welcome. My name is Stephanie Hagstrom, and I'm the Director of Community Development at the Research Data Alliance US. Today's webinar is part of the 2021 webinar series hosted by the RDA US, and this one is presented as a joint effort of RDA Physical Samples Interest Group and the ESIP Physical Samples Cluster Group. The Research Data Alliance is a community of over 12,000 data experts from 145 countries around the globe who collaborate together in building the social and technical bridges to enable open sharing and reuse of data. Members of the RDA come together through self-formed focused groups, working groups, and exploratory interest groups to exchange knowledge, share discoveries, discuss barriers and potential solutions. They also define policies and test standards. Many of the 97 current active groups have produced outputs to enhance and facilitate global data sharing and reuse. The RDA also has over 26 national groups that support initiatives of the members of those regions and provide a platform for those regions to interact on a global level. There are also 4 regional groups of the RDA that provide resources to support and sustain the RDA global operations. The RDA US is one of those recognized regions and is represented by 2,500 plus members who help strengthen the data science infrastructure community. We're excited that we're partnering with ESIP on this project, and we do have Megan Carter here from ESIP. And we'll go ahead and turn it over to her so she can give a little background on ESIP.
Thanks so much, Stephanie. So I'm Megan Carter, I'm ESIP's Community Director, and we too are very excited about this series and about today's webinar in particular. The Earth Science Information Partners, or ESIP, is a community for global earth science data professionals that works to advance the usefulness and impact of data to address pressing global environmental challenges and to fuel new discoveries. And for more than 20 years now, ESIP has helped members of the earth science data community to find each other across traditional boundaries to leverage each other's expertise and to work together on common data challenges and opportunities. ESIP does this by fostering rich collaborative experiences, including 2 annual conferences, the ESIP Lab, which supports the development of innovative applied earth science technologies, and through 30 community-driven virtual collaborations that span a wide range of topics, from more technical topics like machine learning and cloud computing to application areas like agriculture and climate and disasters to physical samples. Anyone is welcome to join in these groups. No RSVP is necessary. Feel free to reach out to me if you have any questions about ESIP or about how to get involved.
Wonderful, thanks, Megan. Before we get started, we have the obligatory housekeeping items, and I'll go over those quickly. We will be recording, as mentioned, this presentation, and I'll be adding those links to the RDA website webinar page probably within a day. And we'll also upload the slides at the end of this presentation. We have allotted time at the end of the webinar for questions, and there are 3 ways that you can interact with the presenters. If you would like to speak and ask a verbal question, please select raised hand and we will unmute you to ask a question directly. The second option is to post a question in the Q&A, and Joan will be monitoring that area and addressing those questions. And we have people assigned to look at chat, so that you can put it in chat, Q&A, or raise your hand. As mentioned previously, the webinar is titled Supporting Interdisciplinary Sample Data Discovery, Integration, and Reuse. It's hosted by the RDA US, and this is the 3rd joint RDA ESIP webinar series highlighting physical samples. I will now turn the screen over to, well, actually, I'm going to just turn the mic over to Joan to introduce the webinar and its presenters. Thanks.
Great, thank you, Stephanie, and thanks again to RDA and ESIP for hosting this 3rd webinar series on physical samples. As mentioned, the series is jointly presented by the 2 groups that are specifically focused on physical samples within RDA and ESIP. My name is Joan Damrow, and I'm involved in both of these groups, and I'll help moderate the session today, and I organize this session. So I did want to bring up that we'll be pausing the webinar series through this year, but you can stay involved through these RDA and ESIP samples groups and check there for information on when the series will resume. So just to give some background on these groups, the ESIP Physical Sample Curation Cluster was initiated pretty recently in 2020, and it aims to enhance the discoverability, access, and use of physical sample collections. The RDA Physical Samples and Collections group was initiated in 2017, and this group aims to facilitate cross-domain exchange on some key issues and recommended practices that are related to the general representation of physical samples and sample collections. So next slide. So I work in community engagement and data standards for ESdive. And this is a data repository for projects that are funded by the US Department of Energy's Environmental System Science program. So I just want to give a little background here because I organized this session today on supporting interdisciplinary sample data discovery, integration, and reuse, primarily so we can all start to hear more about how other communities have begun or have already been supporting interdisciplinary work with samples. So our data repository in particular is working to support scientists in data publication and to develop community standards and tools that supports research that's really interdisciplinary and highly collaborative, often involving large projects with people spanning a wide variety of expertise. So work often involves a variety of different biological and environmental samples, and we're exploring questions related to topics in watershed sciences, terrestrial ecology, and coastal systems. Next slide. So I'm briefly introducing our use cases at DOE to start this webinar to help illustrate why we need to bring together biological and geoscience communities on how we can better represent samples across disciplines. So many of our environmental system science projects are exploring ecological processes and things like biogeochemical responses to contamination, warming, or other disturbances. So a common workflow is to collect a physical sample like soil or water and then send it out for a combination of microbial metagenomic analyses and a variety of physical and chemical analyses. So these are often done across numerous collaborators, labs, and user facilities, and then are often published in different online data systems. So we've begun working through some use cases where we use common persistent identifiers and metadata to more efficiently link and exchange information across these different online data systems that are often currently disconnected. So this has been a fairly long process, and we're still working in a testing and planning phase, but in general, our goal is to make publishing high quality metadata and data for interdisciplinary samples easier. And then to also make interdisciplinary sample tracking more efficient and generally improve our ability for people to find, compile, interpret, and reuse interdisciplinary sample data. Next slide. So we can begin coordinating more across data repositories and other online data systems to more effectively and efficiently use tools like persistent identifiers and common metadata to generally make sample data more fair. And so doing this can help improve the efficiency and accuracy of sample tracking. It can make interdisciplinary sample data more fair, for example, by cross-linking related interdisciplinary data to expand access pathways for data that may exist across a number of different data systems. And generally, using some level of consistent practices across disciplines can help make standards easier to implement and generally improve tools for interdisciplinary publication and access. So we'll be having some more extended discussion on this topic during an associated session at the RDA plenary meeting on November 9th, which is listed here. And we also have a related meeting on November 4th, which is focused on interdisciplinary microbiome data. Next slide. I'm really excited that we have today experts representing different fields of research who have been working on similar issues for a long time now. And I hope by bringing people together here and in future meetings and efforts that you'll hear about today, we can continue to coordinate across disciplines to help make this interdisciplinary sample data more fair. And so with that, I'm happy to introduce our presenters for today's webinar. Our first presenter is Kerstin Lehnert, who many of you know, I'm sure. Kerstin is the Doherty Senior Research Scientist and Director of Geoinformatics Research Group at the Lamont Doherty Earth Observatory at Columbia. So her efforts focus on the development of community-driven data infrastructures for Earth, environmental, and planetary sciences with a focus on observational data generated from physical samples. Our second presenter is Ilene Karsch Mizrachi. Ilene is the program head of Sequence Archives and Submission for the National Center for Biotechnology Information, or NCBI, in the United States. And Ilene has worked at Genbank for more than 25 years. And our third presenter is John Deck. And John is a research programmer at the University of California, Berkeley, working with the Berkeley Natural History Museums, as well as the owner of BioCode, which is a software development company specializing in tracking biological samples. So thank you all again for agreeing to present today. And now I'll turn it over to our first speaker, Kerstin.
All right, just a second. Hopefully I can get this going. Might take a moment for the slide to show up. And so in the meantime, I can say welcome and thanks for the opportunity to present here about two new projects I'm involved with that I'm really excited about. These projects aim to support the transdisciplinary use of natural history material samples and really help to realize a lot of the ideas of sample access, sample discovery, sample interoperability that we've been discussing in these different groups, in the ESIP group and in the RDA group. These projects are really driven by the vision that in order to maximize the substantial value of today's samples for tomorrow's science, we need to really advance the ability of people, of researchers and the general public, anybody to discover, access and use material samples that are generated and studied in cross-disciplinary, both basic and applied scientific research. And to make sure that these material samples are fully integrated into modern day digital and data-driven research. We also need to make sure that connections can be established that are possible now, but to really connect diverse and disparate sample-based observations. The technology is there, but our structures have not supported that so far. As Joan really demonstrated very nicely, there is an increasing need in the sciences to study samples across disciplines. And I've just listed a few examples here. I very much liked Joan's examples. So in all of these cases, be it a soil sample, an ocean water sample, a pottery sample, or a rock sample coming from quite different domains, they are collected for a specific purpose, but they can be used by many other researchers in different domains to extract further knowledge from these samples. And this is really what these projects aim to support. Ultimately, the goal is to create fair samples, meaning that we want samples to be more accessible, openly accessible, and easily discoverable across disciplines. And that requires the samples to be documented with sufficient information that not only provides the ways to find the samples, but also to reuse them based on appropriate information on their provenance. And we want to make sure that the samples are linked to the data so that data generated from samples becomes interoperable and connected, networked, and that it's linked to the interpretations made based on the data generated from these samples and published in the literature. The two fundamental requirements for that is that samples need to have globally unique persistent identifiers and that they have persistent machine readable metadata to which the persistent identifiers resolve. This is the way basically to integrate samples into the digital research data ecosystem. But in order to really create a functional system and a broadly adopted system, the approaches to sample documentation, sample identification, policies around samples for ethical access, ethical distribution, or appropriate citation of samples in the literature really need to be consistent across disciplines. And unfortunately, the status of our sample data ecosystem is not so good. It is very heterogeneous, patchy, and not supporting fair samples. It's fragmented across the many disciplinary communities and also different types of stakeholders, such as museums or academic institutions. There is many different institutional catalogs exist that are not connected. We do have different practices in different domains for unique sample identification, just to name IGSNs, ARCs, DOIs that are all being used, and some they don't match necessarily in all their functionality and capabilities. And we have clearly discipline-specific data and metadata standards that don't match up. An additional problem for fair samples, accessible samples, is that really the majority of samples remain hidden. They are not curated and openly accessible. And this is due to a lack of resources and lack of expertise in researchers, academic institutions, government institutions as well, who are not capable of properly curating these samples. So these two projects that I wanted to introduce, and I know several in the audience have already heard about these projects, and I'm glad that also a number of the collaborators in these projects are in the audience today. These are two separate projects, but that are closely connected in their goal. The iSamples project is sort of the technical side of developments that aims to build the cyberinfrastructure for managing samples across domains. It's funded by NSF, by the Office of Advanced Cyberinfrastructure. The other is sort of the community aspect of building a research coordination network that spans a wide range of domains and provides the scientific vision for working with samples across domains, develops and promotes the best practices and standards, and helps to understand and overcome the social barriers to doing this. And this project is funded by NSF out of the Division of Biological Infrastructure. The team that is behind these two projects overlaps. It is multidisciplinary and multi-institutional and represents really a wide range of domains, including earth sciences, life sciences, genomics, archaeology, indigenous communities, as well as computer sciences, informatics, and persistent identifier services. The approach that both of these projects are taking is to create something that is flexible and scalable to work across domains, both on the technical side as well as in the implementation of standards and policies and so on, and to ensure that it can be broadly adopted and implemented by diverse stakeholders. It recognizes that there are systems, practices already established and deeply embedded in communities that we cannot replace and we don't want to replace. So it's the project specifically built on and want to integrate with these established discipline-specific infrastructures to extend the existing capabilities, but also enhance the consistency of these infrastructures, project systems, and thus augment their impact. The projects want to coordinate as much as at all possible with international standards organizations and infrastructure organizations to, again, ensure that it plugs into existing infrastructures. And both projects aim to work with, promote high-level metadata standards for natural history samples that work across biosciences, geosciences, and archaeology, while also supporting the community-developed, more detailed metadata standards for these specific domains. In order to achieve this, we want to work as closely as possible with interdisciplinary sample-based research communities and seek their guidance. We want to focus on the natural history value chain in sustainability science and engage researchers from diverse disciplines to define the research questions around material samples that would not be possible within a single domain. So just, the overview of these two projects is really high level and basic, and I encourage you to seek further information from us if you're interested. The iSamples project aims to design and develop the cyberinfrastructure for multidisciplinary samples. It will implement 4 initial instances of the iSamples in a box, which is one of the components of this cyber infrastructure, to test use cases that can then, grow adoption in their communities. And in order to do that, again, we want to engage with developers, individual researchers, and international organizations that are concerned with material samples. So just a very brief overview of this cyber infrastructure. It consists of basically 2 components. The iSamples in a box is a standalone software package that interested institutions, organizations can deploy. It creates identifiers and stores associated metadata, and then synchronizes those with iSamples Central, which is a central aggregator of metadata that provides discovery and resolution services with an API as well as an online search interface. The iSamples Central provides a gateway between the iSamples in a box and the different identifier authorities. So nobody who uses an iSamples in a box has to dedicate themselves to one only identifier, but can choose which identifier they want to use.
1 minute, Joan.
Okay, that will be a challenge, but let's go. So we have these 4 different initial implementations in archaeology, in biodiversity, for the geosciences, and one multidisciplinary case, which is the Smithsonian Institution with multidisciplinary collections. iSamples Central's primary component is these resolvable, transformable sample records that are uniquely identified, machine readable, and really provide well-formed context, syntax, and semantics. And in order to do that, the project develops and uses meta vocabularies that are under development. The iSamples the Sampling Nature, RCN, aims to bring together an international interdisciplinary group of PIs engaging across multiple communities, agencies, and so on. And I'm listing the goals of the RCN that I've already mentioned before. The activities are working groups focusing on standards, policy, uncurated sample collections and training materials, and engaging the community through workshops that focus on sustainable development goals of the UN and discussing basically these topics that the working groups are interested in. The deliverables are primarily this extensible community-driven metadata standard and recommendations for its long and short-term and long-term governance, as well as policy recommendations, training materials, and increased awareness in the community about the value of material samples. So here are just a few synopsis items that we clearly recognize science today demands for fair samples, and these fair samples to be discoverable, accessible, and reusable across disciplinary domains. This in turn requires consistent approaches for managing samples in the digital research data ecosystem, and iSamples and the Sampling Nature RCN aim to help in this process to create a discipline agnostic, technical, social, and ethical framework for fair samples across disciplines. So finally, I just invite you to participate, contribute to iSamples, participate in the working groups and workshops of the Sampling Nature RCN. And I'm providing some contact information here. So with that, thank you, and sorry for being a little long. And now I actually need to quit the sharing, sorry.
Yeah, thank you so much, Kerstin. Now we have Ilene, who will discuss how data linkages have made it easier to find, integrate, and use genomics data stored at NCBI and specimens housed in repositories.
Sorry about that. I was talking while muted. Thank you very much for inviting me to participate in today's workshop. I appreciate the opportunity and the opportunity to learn about other disciplines besides genomics, which I am very deeply involved in. So I am the head of the Sequence Archives at NCBI, which is part of the National Library of Medicine in the US. And we archive globally comprehensive scientific record of genomic sequence. We get sequence data from scientists all over the world. We are globally comprehensive in scope and in taxonomic breadth. And we have a broad user community from all disciplines that use our data to make discoveries of their own in very different ways. We manage the intake of data through a number of submission tools. We have analysis tools to analyze the genomic sequence data. And we've been, over the past few years, we've been working to make the data more fair. We are emphasizing providing rich metadata about the samples that are the samples that are being used to generate the data, to provide rich information. I'll get into that a little bit more. We also have, in addition to the archives which house the sequences, we also connect to other resources within NCBI to allow for greater discovery. So we archive data at all different levels. We archive the raw sequence reads that come off the machines. We have variation resource. Then we have resources that archive data that's been analyzed, genome assemblies, gene expression, variation data, and all that data is connected to literature, and the interconnection of all these layers allows one to derive knowledge from the data. Today I'm going to talk a little bit about a couple of the metadata resources that we have within NCBI. There's BioProject, which is a BioProject houses information about an initiative, a sequencing initiative, as well as links to other resources. And it's really an organizer of the initiatives and all the data that we have. So there could be data in disparate resources that are all tied together in an initiative. And it allows you to browse through all our holdings within the resource. We have BioSample, which houses descriptions of the biological source materials that are used in these experiments. Within BioSample, we have a number of different metadata packages to encourage our submitters to provide rich information about the samples. With the NCBI Taxonomy database is another resource that's used worldwide. It's a curated classification nomenclature for all the organisms for which we have sequence data within the INSDC, which is the International Nucleotide Sequence Database Collaboration, which is us, Genbank and SRA at NCBI, as well as ENA at EBI and DDBJ in Japan. We also have a resource, BioCollections, which I'll talk a little bit about, which is metadata about collections, for instance, culture collections, museums, herbaria, and other natural history collections. And not really a resource, but also we have a service called Linkout, where you register URLs for data within your own collections, and just to allow us to point to your resources. And you may, there's a Linkout provider, the person who is a Linkout provider maintains the links, so to different sequence records, to PubMed articles, to taxonomic resources. This is an example of a BioProject, just to show how the linkages are facilitated between the different resources. So for instance, if we have a project that has a title, it'll have, the submitter could include grant information, and for NIH grants, we link back into the NIH granting system, so you can learn a little bit more about the funding initiative that sponsors this project. We link to publications, we link to all the different data types. So here we have some genome assemblies with lots of protein sequences. There are PubMed articles and links to the sample. This is an example of a biosample. Within biosample, we offer submitters the ability to use different metadata packages, and these packages are sometimes organism specific, or they're different data types. So we have, like this, in this case here, we have a model organism package, which has attributes that are appropriate for model organisms or larger animals. We have pathogen packages. This past year, we made a specific package for the SARS coronavirus that's causing the COVID pandemic, with fields that are really appropriate for that, including infection status, whether the individual, the host was vaccinated or not, and other rich information. We also host the Mixis packages from the Genome Standards Consortium. That's a choice that when you submit, you could use that. We work with PHAGE, which is the public health initiative around genomic epidemiology, and have pathogen packages that we've created in conjunction with them. Within a sample, we provide taxonomic information. In this case, it was a bird, and the information is where the data was collected, by whom, when, where it was collected, what kind of sample it was, and we even have a link out to the museum specimen. In this case, it's the Smithsonian Natural History Museum. We also link back to the bioproject, as well as to the data and other samples from within the project. This allows, and by navigating through the information, it allows you to learn more about related organisms, related information. The NCBI taxonomy database is, like I said, it's a curated collection of nomenclature and classifications. We only include names and taxonomic concepts for organisms for which we have sequence data for. So it's not fully comprehensive. There's a lot more taxonomy out there, but we've decided that we would scope it just to what we have data for. So this is an example of a taxonomy page. These are link outs that I mentioned before. So we link to Encyclopedia of Life, the Integrated Taxonomic Information Service, iPhilo, and if there were other link outs for this organism, we would show that here. We also link to the various NCBI resources for which we have data for this one organism. And this is an example of a Genbank record, just to show you how we use links both internally and externally to allow users to navigate through the data. Within the Genbank, okay, within the Genbank record, we link back to the bioproject if you want to learn about the initiative. We link to to PubMed so that you could read the journal article. We link to the specimens with it. If the specimen is in a collection that's in BioCollections and they provide a URL rule for us, we will link directly to the specimen page. So here's an example of also from the Smithsonian. Is a link from here. You could get to the specimen page and learn a little bit more about the specimen. We also collect locality information and GPS coordinates, which will and then link to via the maps to to the region where the sample was taken from. And then the rest of it is the Genbank record. One thing, so when we link to specimens, we use the Darwin core triplet, but would be happy to discuss with iSamples and any other groups. The other standards that are coming about, and we would consider supporting them as long as they are supported by the larger community. And but we are afraid, because there are so many different standards of different types of standard UIDs, that we are waiting to see what comes out to be a more global standard that we would be happy to support. And then I just included the links to our various resources that I discussed today, in case you wanted to look into them further. And there are a lot of people who are involved in putting all these resources together, and the work was supported by the intramural research program at the National Library of Medicine, the NIH. So thank you.
Great, thank you, Ilene. Now we will turn it over to John Deck, who will present on an approach for using ontologies to assist with integrating and linking biodiversity data while inferring new information from source data sets.
Hello, yeah, thank you very much. So this talk is going to be a bit like making sausage, a tour of a sausage factory. We're going to see some nuts and bolts of how things are done, with the goal of illustrating how the system works. The idea here and the motivation for this is that, you know, there's been a lot of great work with ontologies. Usually the ontologies, what you can do with reasoning is limited to, you know, 100 or maybe 1,000 classes. But the goal that we have is to use, how can we use ontologies for large scale data integration and inference. Okay, so I'm going to use the plant phenology work that we did a couple of years ago as an example here. We've since extended this towards vertebrate traits and as well as Superfund, EPA Superfund sites and contaminated samples. But for this case, we're going to look at plant phenology and looking at data sources from the National Phenology Network, the Pan-European Phenology Database, some herbarium specimens, and NEON. All told, we aggregated over 25 million phenology observations. So we start with the ontology itself, and in the ontology has, you can see here, you know, a whole plant, which is defined by PO, which is the plant ontology. And you can see that the phenology observing process here has a specified input of a whole plant. And something, you know, one of our definitions that we have here, unfolded true leaf presence, is a quality of a plant. And the phenology and observing process has specified output, a measurement datum, which is defined by the information and artifact ontology, with data properties of lower count and upper count. You can also see off to the right here that there's some there's a superclass relationships here which will come into play here. So we have defined traits with subclass, superclass relationships defined. All right, so how does the system work? The first thing we do is we assemble data from our input data sources. There's some data alignment that happens, and this is scripting usually done in Python or R, just to get the data formatting correct. And then there's the processing step, which is, there's a tool here, Ontology Data Pipeline, which we can show. This is a tool that's a project on GitHub that runs in a Docker container that anybody can download and use. And it generates an output, and the output could go into a document store or a relational database or a text file. In our data frame, it doesn't really matter. So for the first step, aligning input data sets, we want to create a uniform data structure. So the first record here is an example of a record from the National Phenology Network. The second record here is an example from the Jepson Herbarium. And you can see that there is, things have been standardized here. Some systems might, provide a date. Other systems might provide a day of year. So we want, just to make our computation easier, we're going to put, everything in a day of year. We're going to standardize the latitude and longitude into decimal latitude and longitude. And also we're going to perhaps do a little bit of massaging here in the stage. And the stage is an important data attribute here, which we'll look at, and we can follow this one. The first record here, open flowers, we're going to follow a little bit later on. And just to say that this first record from National Phenology Network, this was a plant that someone noticed had some open flowers, and lower count of 1 indicates that they counted at least 1 open flower on this particular plant. So we dive into the functions of the pipeline. The first thing we do is convert to RDF triples, which is a format of subject, predicate, object. And this is a bit of a data formatting change. And we also map project terms to ontology terms. We assign unique identifiers where applicable. For all instances of classes need to have their own identifier, and we create relationships. So there's some configuration files and configuration steps which we need to define what are the ontology entities that we use or that we're assigning here. So if we look at row 4 here, measurement datum is the name of of an entity. And here's the definition from the IAO, the Information Artifact Ontology. And there's a definition of whole plant, which is defined by the plant ontology. So these are a few of the entities that we're going to be looking at. And then we map the field names to ontology terms. So this is where we have, a data integration step. You have a field that comes from the source data sets, for instance, open flowers, in parentheses, lilac. And we're mapping this to open flower presence. So all of our data sources that have all of the incoming data, we need to map these into the defined by column, and those things in between curly braces correspond to RDFS labels, which are defined in the ontology. So this is kind of the nuts and bolts and the part where we need to consult with botanists and consult with subject matter experts. What do you mean by open flowers lilac? And you say, well, they can tell us, by the definitions that are in the ontology, what they are. So once we're done with the triplifying step, we come up with a sample outfit that looks like this. Looks like it's a bit of code here. This is the data structure. We have an instance of a trait here, so you can see that there's an arc identifier that is a type A, open flower presence. So we haven't asserted anything. We haven't even asserted that a flower even exists, but we have asserted that someone looked for the presence of an open flower. And has a specified output of an instance of a measurement datum. And if you look at the identifier for the measurement datum, here's the definition for the measurement datum. It says, this instance is a measurement data, and it has a lower count of 1. This is cool. This allows us to move on to the next step, which is the reasoning. The reasoner, we've used the robot tool. And actually, when we developed this, we developed our own tool. And some of the code from our own tool went into the development of the robot tool. This was Brian Stuckey had a tool called Ontopilot, which then got sucked into robot. So this is a newer tool, which is a Java program where you can specify different types of reasoners to use. And there's a selection of 5 or 6 different reasoners. We chose the EL reasoner, which does the type of reasoning that we want to do and is pretty fast. So we end up with a reasoned output of, we have an instance of a trait here at the top, which is A, open flower presence. We still haven't asserted that anything's present, but on the next line you see now, wait a second, we're assuming that open flowers are present. And this happened because we had a minimum number of flowers that were observed, and we can say open flowers are present. We can also say that since open flowers is a subclass of floral structures, that floral structures are also present. And floral structures is a subclass of reproductive structures are present, then they're also present as well too. So you can see we're starting to infer a lot of new information here in the reasoned output. Okay, so you're left with this RDF triples. Most people don't like to look at RDF triples, and most systems are pretty slow on doing that. So we could, there's a final step of formatting, which basically kicks the data out into a format that looks a lot like our starting point, but you can see over on the right here that there's a bunch of inferred stages, which are now put in here, that now we're saying, yes, flowers are definitely present. When in the first stage, we just said we're looking at open flowers with a lower count of 1. So just looking at the system, just to see, what is possible? How much data can we run through these systems, for reasoning? I ran this data set, 25 million records on my MacBook Pro, took about 8 hours. And the final format can be easily loaded into any type of relational database or document store. So if you're interested in, query times of, on the level of, 1 or 2 seconds, and a lot of this can be queried very quickly there.
1 minute.
So on our final output, we have some sausage, which is nice and tasty. We just took a tour of our sausage factory. If you want to forget about all that, you can just, focus on the end product, your inferred data source. You have, and there's a link to the some of the source code, so you can try this on your own data sets as well. That's all.
Okay, thank you, John. So now we have just about 8 minutes left for our question and answer period. And so I'm going to start with some of the questions that are open in the Q&A.
And the panelists want to turn on their cameras.
Yeah, so the first question I have is from Andrew Bentley. And that is asking whether, this is for Ilene, whether link out and structured voucher systems will be ported across to SRA, and whether NCBI will adopt unique identifiers that allow linking of genetic sequence information with voucher and tissue information in natural history collections.
Okay, so I know Andrew sent me a chat that he left, but I will follow up with him by email. So SRA houses all the source metadata in biosamples. So we already use the structured vouchers within the biosample. Link out, I am not sure about. It should be available, but if it's not, I will look into that. And the rest of the question was the unique identifiers. We are looking into adopting a system of unique identifiers. The question becomes which of the many systems we should adopt and support.
Okay, thank you. I'll move on to the next question from Anita, which is also about link outs. Is there a way to get some text beyond the name of the project into the link out text? She
that
said,
I would have to follow. I'll follow up on that one as well. I link out. I'm not as familiar as link out as some of the other resources. And Andrew's other question, I will deal with.
yeah, and so yeah, if we have answers to questions afterwards, we can send a follow-up message to everyone answering some of these questions that we don't get to as well. Okay, the next question is, if we allow curation of sequences by museum staff or vouchers are from our collections. I'm not sure that I.
Yeah, that's Andrew Bentley. I will follow up with him offline. We've worked with him in the past. We do allow some updates from museum staff because of the way the data is submitted. The submitter is the ultimate owner of the data, so that sometimes makes it difficult, even if we want to be helpful to the museum community. But we'll look for ways to help.
Okay. So there was a follow-up question from Anita on link outs as well, but it sounds like you may be also answering that later. Let's see. I think those are the questions in the current Q&A.
There are 4 that are answered previously,
Oh yeah.
typed, but not verbally
Okay, let's go
discussed.
through these verbally as well. So the first one was, how do natural history museum collections fit into the iSamples model? And John and Kerstin answered that, but if you could just discuss that here now as well.
John, you want to go?
Sure. Yeah. So natural history collections are, as far as iSamples are concerned, is very similar to looking at rocks or archaeological artifacts. They're, at the core samples. There's a bit of different descriptive metadata that goes along with a biological sample that would be found in a natural history museum collection. And one thing that might be a bit different, though, with natural history museum collections is that there's systems or archives are there's certain flavors of them, Arctos or Specify or KEMU, that people use, which I believe that our interactions with the museum collections would be interactions with those systems on the back end.
Yeah, it is, as I put in the answer, it's a specific deliverable in the developments is to create these adapters between local data sample management systems and the iSamples instance. And the best use case here will be the Smithsonian Institution, where we're gonna have an iSamples in a box instance to do the connection to iSamples Central, but the Smithsonian is obviously not going to change their sample management system, continuing to use what's there, and it's a pipeline of metadata basically.
Okay, following on iSamples, do you want to say anything now about the use statistics that are available for iSamples, or that will be available?
Yeah, it's my fault. I didn't quite give everybody a feeling of where we are with iSamples. iSamples started slightly more than a year ago, 15 months or so, and we are still deep in development. The first prototype of the iSamples in a box and iSamples Central are expected in about a year. That's when we're going to start building the use case in deployments and testing. So there is no adoption statistics yet. But the use, obviously, I mean, I wasn't quite sure if I understood the question entirely. It could also pertain to, is iSamples capturing statistics of use of samples? And that should clearly happen. That would be part of you know, the function to really give operators a way and users a way to check how often citations of samples have appeared. Once that is fully implemented, the links between publications and samples are fully implemented in iSamples Central.
Okay, now we're at 2 o'clock, so I think Stephanie is going to close out really quick. Sorry about that.
That's okay. Let me just get my screen up. I just want to thank everyone. It's 2 o'clock. I'm sorry, we're going to go over just a few minutes, 1 minute. I want to thank all the speakers, Joan for leading the webinar, Megan for helping behind the scenes, and all the questions. I'll grab all the questions, and if any weren't answered, we'll go ahead and send them to you. Just wanted to give you a heads up. We have another webinar in October, and this one is geared towards those who have outputs or research that they want to get out attention. How do you market these things? So it's how to get attention for research project outputs. And that one's on October 21. We have the 18th Research Data Alliance Virtual Plenary coming up November 3 through 11. There is one more webinar on navigating all things RDA, website, plenaries, and more, October 26th. And if you haven't joined RDA, we hope that you do. Here's some emails and websites that you can go to get involved. On behalf of the RDA and the RDA US, we thank you so much for joining us today, and apologize for going over a little bit. And it was a really great, clearly very interesting topic that you all need a little more time. We all need more time from all of you. So thank you all so much. Bye-bye.